Hi, everybody. This is the first video for the third week. We're going to start getting into the structure of viruses. So I'm going to bring us to our lecture. This one is module 3. We're going to be looking at virus structure. The learning objectives for this particular mini lecture is that you should be able to, at the end of this, draw a basic virion structure. I'll draw one on my whiteboard so that you kind of get an idea of the basics of it. I also want you to be able to describe the genome, capsid, envelope, and spike proteins. You should be able to describe the functions of each of these structures as well. The vocabulary for this particular mini lecture include virion, genome, capsid, capsomeres, icosahedral, helical, complex, envelope, naked, and spike proteins. The basic viral structure is a nucleic acid center, so the DNA or RNA that has the information to make that virus enclosed within a protein shell that we refer to as a capsid. Now on this one, you can see that they're showing DNA inside of the virus or the virion. This could also be RNA. There are viruses that their genome is RNA or ribonucleic acid instead of deoxyribonucleic acid. The outside of it then is protected with this protein capsid made up of repeating capsomeres. And in this one, it's not technically the basic viral structure, but we could also have these spike proteins that are made up of sugar and proteins called glycoproteins. For me, when I think about this, and I will often draw this up on the whiteboard, what we often think about with it is that it's just that outer shape. Let me bring up my whiteboard for you. We can think about as the most basic shape, so basic virion. Remember, a virion is the term for one individual infectious particle. So the way that I generally draw it is I draw my capsid, which is this protein shell around it. So this is my capsid. And then internal to that is what I would say is the nucleic acids or genetic material. So this is genetic information. And that can be either DNA or RNA. Okay, so if I ask you to draw the basic, that's what I would expect to see. Keep in mind that that capsid would be made up of capsomeres, which are just individual repeating proteins. So you might think of it as one side is a capsid, a capsomere, 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 all of them together are a capsid. This is giving you a picture of kind of what these capsids might look like. So again, these are just some other drawings of it. You can see that the capsid would be this outer part out here in gold. It's showing you the spike protein sticking out kind of in purple. And inside, we would expect to see the genomic material or the DNA. It's showing that there maybe would be a protein. Sometimes some viruses carry their own proteins with them. When we put the genetic information and the capsid together, we can refer to that as a nucleocapsid. So that consists of the capsid and the genome. This is just showing another picture. They can have different shapes, but hopefully that gives you a good idea of what a basic structure for a virus is. So to give a quick review, a virion is a single virus particle and the genome is enclosed within the capsid. Capsids are protein shells that package and protect that genome. It makes up the bulk or the majority of the virion's mass, and it's made up of those subunits called capsomeres. Most animal viruses are one of two shapes. They're either what are called helical, which would look like kind of a tube of a capsid with the genetic information inside, or icosahedral capsids. The capsid sort of looks like a geometrical shape, very much like what I draw as my basic structure. So again, the helical capsids look like the hollow tube. Icosahedral are those three-dimensional polygons. There is some types of viruses that will deviate from these two capsid structures. And if it's not one of these two, either the icosahedral or the helical, we classify those capsids as what are called complex capsids. Just reviewing from what we talked about earlier, there are certain viruses that affect or infect bacteria only, and we refer to these as bacteriophages, so viruses that infect bacteria. And these are viruses that will exhibit what we call a complex capsid structure. And these ones have usually a capsid that still looks like an icosahedral shape, but they have these other associated parts to them that allow them to actually land on a bacterial cell and inject their genome into the target cell. So they look, to me, a little bit like a spaceship. I think they're really cool, and I think I have a figure in just a minute. Yeah, so a virion, again, that single virus particle, the genome enclosed in a capsid. This is giving you a view of these different shapes. So we have the icosahedral here, that's this geometrical shape. We have a helical, as I said, that looks like a hollow tube. And then those bacteriophage can be a complex. So you can see up at the top, it still has that icosahedral shape to the capsid with the genome inside. But now it's got this long tube and kind of these landing legs that allow it to attach to a bacterial cell and inject that genome into it. And we'll get into that as we move through the replication life cycles of different types of viruses later in this particular module. Some animal virus have what are called envelopes. So SARS-CoV-2, what causes the coronavirus or COVID-19, has an enveloped virus. And what that means is that it has this lipid or fat-based envelope that will surround the exterior of the capsid, so around the outside of it. And it actually steals this from the host cell. So when it's made in the host cell and it goes to exit the host cell, it will actually take part of that membrane with it that was the host cell membrane that you learned about in the second week of this class. If it takes that with it, it actually has kind of this extra layer of fat around it. Some viruses, though, do not take that as they leave. And so we call these naked or non-enveloped viruses. And these ones, rather than kind of budding off from the host cell, they will actually break apart the cell membrane at the spot where they're exiting the host cell, and they won't take that with it. Interestingly enough, enveloped viruses are actually much more susceptible to things like soap and water than a naked virus would be, because that soap and water can break down that fat-based envelope around it. And that's why just washing your hands with soap and water will actually inactivate any of the enveloped viruses. Animal viruses may be either enveloped or naked. Some are enveloped, like SARS-CoV-2, and others are naked, and we'll talk about some of them as we move through the class. Naked ones, though, are harder to control or harder to inactivate than enveloped ones. I do want to make a small note, though, that bacteriophages, which, if we go back, are these complex virions. These actually always, when they leave a bacterial cell, they're always going to break open that bacterial cell and kill it when they leave. So they don't take a cell membrane with them, and therefore they are always considered naked. I did show you in one of the first pictures that there can be these exterior proteins that are sticking out, and these are called viral spikes. So most viruses have spikes that will stick out from either the viral capsid if it's a naked virus, or from that membrane, that envelope, if it's an enveloped virus. Those glycoprotein extensions, their whole job is actually to act like a key to unlock the cell to gain entry into the cell. So that's a really important part of these viruses, is they actually trick the cell into thinking that they know kind of the secret password to get into the cell. And I have in the picture over here, this is an example of SARS-CoV-2, so what causes COVID-19. And it has these spike proteins. This is the virus, right? It has its genome inside in kind of this blue. It has the capsid as kind of this blue part sticking out around it. And then these pink parts are those spike proteins. Those spike proteins can actually attach to the outside. So this is inside a host cell. They can attach to these receptors called ACE2 receptors that are on much of our respiratory cells. When this attaches, these spike proteins attach to that ACE2 receptor, the host cell gets tricked and thinks, oh, I should bring this in. And so it will actually take the cell in, and this is where an infection would start. So these spike proteins are really important for a virus to get into a host cell. I'm going to pause there. You can pause the video and answer these questions before you come back and I go over them. Let's go ahead and go through these questions. The first question is, what are the units called that make up the capsids? That's capsomeres. Number 2, explain where enveloped viruses pick up the membrane from. They pick up the membrane from the cell membrane that is surrounding that host cell. And in some cases, they can also pick it up from the nucleus within the cell. What is the function of protein spikes? To bind the host cell receptors and gain entry into the cell for the virus. What is always found in the capsid? That's going to be the genome. If you have any questions about this, please let me know, and I'll see you in the next mini lecture.